Good morning, everyone. Here we are in this first workshop on seed priming for a sustainable agriculture. And my name is Vicente Arbona, and I'm together with Alma Balestrazzi from Universitat de Pavia. We are coordinators of two PRIMA projects, and we are promoting this workshop to present the breakthrough in different seed priming technologies and also the underlying plant biology. We are here in this hybrid event in Universitat Jaume and Universitat de Pavia, and we have different presenters live here in on-site and also online. I will now introduce my colleagues, Dr. Alma Balestrazzi from Universitat de Pavia, and also we'd like to introduce Fabrice Interesangle from as a PRIMA officer, project officer, that will be here with us today. So I would like to first thank Fabrice Interesangle for being here, and I would like him if he wants to say some words. Thank you. Good morning.
Thank you very much, Vincent. Thank you for inviting me to this event. I'm happy to be here. Good morning to everyone, of course. It's a pleasure for me in reality to be here because, as you said, you are coordinator of two PRIMA projects. So you are organizing this workshop together. And in PRIMA, we are pushing for this synergy. We are pushing for this collaborative work. So I'm very happy to see that your two projects are collaborating as you are working on very similar aspect of seed priming or trying to find the best microorganism that can help us in the future for to have a better agriculture, sustainable agriculture. So I'm really happy to see that there is this collaboration, that there is this mentality of this spirit of PRIMA that is really to share and to go on, not to repeat each one in his country, what the other are doing, but to put together all the knowledge you have and to go further to help the region. So thank you very much. And I know this is also a first step for you because you have also another event organized with 3 other PRIMA projects, so 5 PRIMA projects in total. So I congratulate you also in advance for that because it's a very good news and we are always happy to see that. So I'm really wishing you the best for this journey. You have a very nice agenda. I really hope you will benefit for this cross-fertilization, let's say, between projects. And so I just wish you the best and thank you again for inviting me to this event.
Thank you very much for your words. Really encouraging. Now I will introduce Dr. Alma Balestrazzi. She's coordinated the Benefit Med PRIMA project, and she will give us a very brief outline of this huge and very interesting project. Thank you very much.
Thank you, Vincent. Thank you, Fabrice. Just let me open and share the presentation, brief presentation. So, just need to catch the PPT.
Non è condiviso lo schermo, giusto?
Can you see it?
You have to share.
Bisogna condividere lo schermo.
Allora, just a minute. Okay. That's
Schermo intero.
okay, and this is finestra. Okay, now it's shared. Oh,
Back in
no.
Ok.
Just
No. Interrompo.
Sorry, it happens to me the same problem. I just tried yesterday. Maybe it's better if you open just as a window. So then you can project better because it's going to do this loop of the full screen. So I don't know, this is just an advice.
That's better.
Okay.
Thank you, Cinzia. Yes.
No problem.
Ok, mettiamo anche sotto. Schermo intero.
Per modalità presentazione.
Okay, can you see it now?
Yes.
Yes, so a few words to introduce the Benefit Med project. This is a project in which, for example, in which the main goal is to develop tailored seed priming protocols to boost the germination performance of selected legume species that are neglected species, orphan legumes as they are called. They have been discarded from the point of view of breeding, of molecular investigation, but
Sorry. Just one thing. Could you please put the whole screen, full screen, please?
Yeah, the full screen is already there for us.
In PowerPoint, you have to launch the presentation.
Okay, just a moment.
Oh, on your right, that one.
Is it okay now? Because we see the whole screen, but I don't see other. Yeah. Can you tell me, please?
Well, what happened to me was that keeping the window of the meeting, and then you launch the presentation, and then it's coming out. But I mean, you will not see the people, at least what I could see yesterday. I mean, it's when I launched the presentation. So you keep just the window with the, at the beginning you upload the window, okay, like this. Then you keep a window open and then you click on your computer PPT and then you open the full PPT. If you want, I can try with mine just to see what you, I don't know if I'm, se no Alma ti parlo in italiano, if it's more clear. Okay. So do you want that I try with mine? So
We'll follow your.
Because
Okay.
it was working before, so.
yeah. It's some problem with me. I don't use it very often. So yesterday I tried and it was like a window. You keep the window of meeting, you keep sharing. Afterwards, you launch the presentation for the computer and then it's open also in your meet. But you don't see the people, so I don't know if it's just a matter of settings or.
Let's see the window. Okay, just vediamo.
No, it's always like this.
You see now the presentation.
Not in full screen.
Okay, and now if we go to the full screen,
Let me check.
like this, is it okay now?
Not in Meet.
Ah, I just called.
It's okay. I mean, you can continue with this. Let's do it like this. Don't worry. Even if it's small, you can continue like this. If you cannot put the full screen, continue like this.
Yeah, it's okay like this, Vicente. Sorry for this inconvenience. Okay. Okay, so.
Semplicemente non sappiamo come avviare la nostra nell'intero Google Meet. Perché ho provato a condividere via schermo intero, ma era una cosa dei...
It's okay like this?
It's okay, we can see it
Okay,
perfectly, don't worry.
okay, we can move on. Again, let's start again. Let me say a few words about the Benefit Med project. This is a project in which we are developing tailored seed priming protocols to boost the germination performance of these orphan legume species. These are highly resilient species because they have so far not very much used for breeding, and so they have retained highly high stress tolerance trait, as well as high nutritional value. So what we want to do, and sì, qua. Questo. Oh mamma mia, non va. Oh mamma. Sorry, I'm trying to. Chiama Claudio. So basically, what we want to do with Benefit Med is to develop a new sustainable, smart agri-food system that can be used, applied by the population, the climate vulnerable populations of the Mediterranean area. And Benefit Med is a consortium that covers 11 partners around the Mediterranean basin. And as you will see in one of the next slides, we move from Germany to France, Portugal, Morocco, Algeria, and Tunisia, and then to Greece and to Italy. And basically, this is a highly multidisciplinary project because we move from the bench to the field. At the bench level, we have developed and validated these tailored seed priming protocols. So then the treated seeds were also tested at the field level. We have several pilot sites with field trials that have been already closed with promising results. At the same time, we are monitoring the system from different point of view. So there are partners specialized in climate science. They are monitoring the climate change events along the Mediterranean basin. There are partners experts in life cycle assessment. So they are trying to evaluate the real sustainability, the real impact of our agro system. And again, we are also setting a database that will gather all the existing information produced by the project concerning these species. If I can move, okay, please, Claudio, can you help me just to avoid? No, it's blocked. Anyway, just to summarize the many activities that we have so far done, lots of training and dissemination and communication activities with several workshops. One of them was held in February concerning the role of women in science. And well, what can I say is that the Benefit Med Consortium is an excellent team. We are working together in harmony, and it's really for me an honor to lead this consortium. So sorry that maybe I was not able to show the nice picture, but they will come back along the workshop, in the day. So.
Thank you very much. Thank you very much. That's technical problems we always have.
Yeah, we'll solve.
Now we'll open my presentation. So we have it in the here. I don't know if you can see the presentation. Okay, so this is our PRIMA project, the optimal usage of natural product and biological priming agents to improve resilience of agrosystems to climate change, which has as a main objective to provide the agricultural sector with a cost-efficient seed priming technology based on the combination of natural compounds and microorganisms that's aimed to improve crop performance and productivity under adverse environmental conditions, contributing to a sustainable agriculture. So essentially is to have a tool that allows agronomists and agricultural producers to have more yield under climate change conditions. So the consortium is constituted by 9 different partners from all the Mediterranean area, including Italy, of course, Turkey, Cyprus, Greece, and also Spain and Morocco. And this is the organization of the project in 5 different work packages. Besides project management, we also had a work package oriented to all the potentially transferable results of the project, to have a particular work package aimed to have all these results and manage the intellectual property rights, and of course a work package in which we developed all the basic science underlying the seed priming technology, and also a work package in which we had to check this technology outside the laboratory, under field and also greenhouse production, since this is oriented to horticultural production, and the greenhouse production of horticultural crops is very important in Europe. And finally, a work package in which the developed technology at laboratory scale is brought to a higher degree of technological maturity, which is high throughput production and industrial production of prime seeds. We also organized several workshops here in our university to show the different strategies for seed priming and also implant priming and the underlying biological mechanisms. We have also boosted different outreach activities. The first and probably the most direct one is the website, which is already running since 2021, and also a highly active Twitter account that can be visited anytime to see the news on our current running project. Also, we have organized several knowledge and secondment activities. The most recent between the Cyprus University and the IPSP in Italy, and these are some summary of the different activities developed. So the checking of the potential priming agents under control conditions, followed by those that really work. We wanted to know the underlying mechanism, so that's where we put all our efforts to understand the biology underlying the improved effect provided by seed priming. And also, we checked other possibilities besides the one we developed within the project. So in collaboration with other different groups, we also checked different combinations of natural products and microorganisms to deliver similar, or if not enhanced, ability to withstand abiotic conditions. And that's pretty much the outline of the OPTIMUS Prime. So I think we can move to the next section, and the next speaker is Dr. Eduardo Bueso from Universitat Politècnica de Valencia. We are neighbors, and he will give a very interesting talk about his work on seed biology. The floor is yours.
Okay, hello to everybody. My name is Eduardo Bueso. I work in Politècnica University in Valencia as professor, and also I'm researcher in a center in the same university. The center is called am-3pm, where we are doing molecular plant biology. Specifically, I work in seed longevity for 20 years now, where we got with blood, sweat and tears some projects, 3 projects specifically. One is current now, and also we have 12 manuscripts. Thanks to the organizers to give me the opportunity, to trust on me to give a talk. And also I'm very happy to have colleagues working now in seed viability, seed longevity, because I felt alone in this field for a long time. So, in this talk, I will try to summarize the most important main mechanisms in orthodox seeds related to seed longevity. Firstly, I start with the controversial definition of seed longevity, because the most accepted is, as you see in the slide, is time interval from maturation until the seed has lost the capability to germinate. This is very controversial for me after thinking about this, because you know at the end of the maturations we have a period that is dormant. The seed is dormant, so cannot germinate. So it's, for me, it's I'm thinking this is a real good time point to start to think that this is longevity, even if the seed cannot germinate. And also, what is germination? Germination, for me, it's maybe root emergence, root extractions, but sometimes, even if seed can germinate, cannot develop, cannot establish a seedling, cannot develop a full plant. So this is open question to think about it. For the beginners in the field, I have to say that you have to be very careful with this feature, because longevity presents a sigmoidal curve. What does it mean? This is that maybe you have a seed batch that is germinating fully, but in some weeks can stop to do it completely. This that I show in this graph, this reflects that a normal distribution where almost all the seeds stop to germinate at the same time. So take care about that. More controversial is how to measure seed viability. For 20 years, I was fighting with the referees what is the proper method to measure seed viability or seed longevity. We have, in this moment, at the moment, I think the 4 main methods to evaluate the seed longevity are these ones. Natural aging. This is not very practical method, because you know the projects are 3 years usually, so it's when you start the project, you try to age the seeds. At the end of the project, you don't age the seeds. This also has a problematic for me, because usually the seeds are conserved in germoplants bank in very low temperature and humidity. But for me, we have to think This is real natural conditions where rains, there are high humidity, sometimes a peak of temperature we have to think about. Also, we have 3 artificial methods. The most used are accelerating aging, that is the fastest method, where usually the seeds are imbedded in water, and we apply also high temperature to age very fast. This has a very critical points, because sometimes the referees or the community, the scientific community, say that it's incorporated or it's we have hypoxia stress because oxygen cannot enter in the seed, or maybe in a lower manner. And also it's very critical because when you apply water or high humidity, the glassy state of the seed, that after we will talk more about it, is disturbed. The most accepted method, I think, is the controlled deterioration treatment, where high humidity is applied, but never disturbing glassy state, and also high temperature. Sometimes too high temperature is applied. And other one that is also very used for specific researchers are the seed copes elevated pressure of oxygen or nitrogen. For me and for other scientifics in the field, we think that this is not good method because the high pressure could disturb cellular membranes or could produce fractures or fissures in seed coat or endosperm cuticle. Also, two things about it, two things to think about it, is that for me, high temperature is we cannot apply to age seeds because it's producing a fast protein denaturation that is not occurring usually with the aging. And also, at what point is disturbed, at what point of moisture is disturbs the glassy states? It's very studied by Boiting and Olivier Le Prince in France for Arabidopsis, but for other species it's not so clear at what moisture or humidity is disturbed. In the lab, we were very worried about this, how to measure seed longevity, how to apply methodology. So we have this paper where we perform a very nice experiment where we try to correlate the 3 methods of accelerating aging. This is applying elevated pressure of oxygen. This is CDT, controlled deterioration treatment, and accelerating aging treatment. We got 270 ecotypes of the model plant Arabidopsis thaliana, and as you can see, the correlation between these 3 methods and germination natural, the most proper method to use is CDT, because it's the most correlated. And it means that at least in Arabidopsis, we don't know in other species, apply high humidity and high temperature, but not very high humidity, don't disturb the glassy state is the best method. And what is the glassy state so important for our field, to study seed longevity? Glassy state it is the process where the seed lost a lot of volume, exactly of water, of loss of relative humidity, but it is very important process or essential process for seeds to tolerate desiccation. The seeds, thanks to the accumulation of LEA, soluble sugars, and heat shock proteins, can survive to these desiccations. This it's forming a glass when cytoplasm solidifies. This is basic because chemical reactions, oxidative chemical reactions, are slow, and with this process, life of the seed is extended. So it's very important to take it in account this process in the seed. As I told you before, I'm going to talk about. I'm trying to summarize all the mechanisms that are published in seed longevity in the literature. spending a little more time with my publication of my lab. So, but one thing is very clear of all the publication is that seed stops to germinate because it's copying the oxidative stress coming from our atmosphere. This oxidative stress is producing aging in the embryo that finally cannot develop. So the plants develop several mechanisms to cope these conditions, but we can classify in four items. Those mechanisms that permits proper seed maturation, I mean, that produce that mechanisms that allow that desiccation, that the tolerance to desiccations, those mechanisms that repairs a misfolded protein that is produced for oxidative stress, combating oxidative stress mechanism that helps to maintain the normal state of DNA and lipids and proteins. And for me, the most important, because I was very relating with this kind of mechanism, is the prevention of oxidative stress, usually because of seed coat. Here I present very summary the mechanism that permits proper seed maturations. This is very important. This is a very old manuscript or publication at the beginning, 20 years ago. Not a lot of publication we could find in the literature, but one of these was the perfect coordination between these transcription factors, leg2, AB3, FUS3, and leg1. Here I present you the connection between these transcription factors and other genes very important, synthesizing different hormones as auxin, gibberellins, and abscisic acid. We can find the JACA genes, the NCAD to produce ABA, and the gibberellin oxidase 3. In the graph, I show you the most important hormones for the maturation and develop of the seed are GA, very important for coat to form the seed coat, especially the gene GA3OX3, and also ABI, that it's very is increased a lot when the seed is desiccated, and after is going down when germination starts. Okay, in this slide I present the three main mechanisms to misfold protein, to repair misfold protein in seed that is published. We have in the first one, in first position, is the PIM1. This is a protein repair, so aspartyl methyltransferase. Okay, so consequence of oxidative stress, aspartic, it's oxidase to isoaspartyl, and this protein, what it's doing is removing or methylating this residue that is very reactive, and is producing this this residue that is not so reactive because of the addition of the methyl. The second one is published by our team. That's we isolated a double mutant, ROF1, ROF2. These ROF genes are peptidyl prolyl 3-trans isomerase that do the rotation of a peptide bond, preceding always a proline. This double mutant we found in the lab that is very sensitive to seed aging, artificial aging. And finally, we have another work that it was published by another Spanish researcher, Juan Jordano. It's a very old paper as well, that Giordano introduced the Hitchcock factor from Elianthus annum, Hitchcock factor A9, to Arabidopsis, and could isolate plants or seeds that are very resistant to accelerating aging in seeds. Yeah, about combating oxidative stress, one of the main targets of oxidative stress in seed is DNA. Here we find three main mechanisms. First one is a publication of Wanda Waterworth. I think is in the audience, or we'll talk this afternoon. I don't know. It's she described the DNA, the mechanism of DNA ligase 6 and 4 to repair seed DNA in seed. I'm not so brave to talk about this work, so we will pass to because she's here. The second one is another mechanism that repairs the oxidation of the oxyguanosines. When the oxyguanosine is oxidated, is forming this new compound, 8-oxo the oxyguanosines in DNA. So to remove this kind of base that it's this is pairing with adenine, the seed or the plant or the cell has the mechanisms to induce this enzyme, this DNA glycosylase AP liase, that is removing this radical and starts to form a new chain of DNA without mutations. The last one mechanism to repair DNA in seed that is published is PARP3. That is it's a polymerase that repairs the DNA breaks or DNA damage through a mechanism that is called pyrylation. That is the addition of poly-ADP ribose chains in the DNA. Yeah, not only DNA is oxidated because of the atmosphere of reactive oxygen species, we have protein oxidation. Here the main mechanism to cope the protein oxidation is the induction of methionine sulfoxylase reductase. As you see, here methionine is usually oxidized to methionine sulfoxyl, and with this enzyme we can recycle and we can get a new good one, methionine. The other important issue is the lipid peroxidation that is produced. This is very old paper as well, manuscript, where here we can see the function of tocopherol removing peroxyl radicals and forming tocopheroxyl radical, and that's way we can eliminate a big part of the peroxyl radicals. Okay, here now in the second part of my talk, I will talk about our work in the field, our contributions. I started 20 years ago with the PhD in a very difficult project and new for the lab in Ramon Serrano lab, where our aim or our goal was to discover new genes and mechanisms involved in seed longevity. We use the accelerating aging method, CDT and AA, because I told you before, the PhD is 3-4 years, and we don't have time usually to age in a natural way. And the screening, we did a screening with a collection Arabidopsis mutant generated by activation tagging. We use this plasmid that allow us to find or to isolate gain-of-function mutants, and it's very easy to localize or to place the mutation through the method as plasmid rescue. Okay, this is the typical scheme of isolation mutant. We do 2 rounds of selections to find very germinability mutants with a lot of germinability. And after, we do the plasmid rescue, check gene expression of the genes close to the mutations, Another very important method is to co-segregation analysis to be sure that the mutation, the integration of the DNA is co-segregating or correlating with the phenotype. The funny thing of this screening is all the mutants that we isolated were gain-of-function mutant lines, and all were transcription factors related to seed coat development. That's why, for me, prevention of oxidative stress is a very important issue to cope oxidative stress in seed. We validate our methodology with the mutants, with another co-segregation correlation analysis between the methods, artificial methods to age and natural aging of the 4 main mutants. And also, we force by referees to demonstrate that our methodology is not producing a secondary dormancy that also happen when stress is applied. Here, we demonstrate that our method is not producing increase of the content of ABA. Well, we published 3 or 4 articles with the isolation and characterization of the mutants, but we went further because we wanted to know the molecular mechanisms involved of these transcription factors I told you before. So in the next publications, we go through the molecular mechanism. For example, COG1 was one of the first transcription factors that we isolate involved in seed longevity, and in these publications, this COG1 was previously published. It was a transcription factor related to light signaling, but also we demonstrate the high expression that shows during the seed coat development, as you can see. Also, with the RNA-seq in the overexpression of the mutant, and we could find a lot of peroxidases that were induced in this mutant. So next step was to check the expression of these peroxidases in seed coat, and we showed that the peroxidase 2, 25, and 72 were very expressed in seed coat. So we go for genetics. We got the double and triple mutant of this peroxidase, and we could check that the double mutant loss of function were very sensitive to accelerating aging methods. After using TEM microscopy, we could see the defects that represents these mutants in peroxidase in the formation of the seed coat. As well, using tetrazolium salts, we could check that the high permeability of the seed coat that it presents. As more red is more permeable. This is wild type, and this is double and triple mutant with high permeability. But it's provoking the rapid oxidation, the fast oxidation of the embryo and cellular components. Finally, we demonstrate that these peroxidase are very important to the polymerization of the aromatic part of suberin in the seed coat. Also, these mutants presents lower lignin content in the seed coat. Another very important for us publication in New Phytologies was the description of homeobox transcription factor very conserved among angiosperms. Here I present the natural aging, the germination after natural aging of several months. Here you see the wild type, and here the gain of function of the transcription factor, and here the big phenotype, the very sensitive phenotype of the double mutant 22-25. 22 is the homologue of 25. And also in this mutant, we play with the overexpression and the loss of function double mutant, and we could check how is involved this transcription factor in the develop on the accumulation of lipid polyesters in the suberin layer that is here in the palisade layer below the epidermis, and in the formation of the cuticle. This double mutant has thinner seed coat, less suberin, and less cutting. Here, with a collaboration with Isabel Molina in Algoma University, we determined that the HB25 is directly related to the synthesis of decarboxylic acids that are essential for lipid polyester composition. And even we performed a very new experiment, was a chip-seq during seed development, and we could find that the HB25 is directly binding to the promoter of genes as LAX2 that encodes an enzyme that catalyzes the activation of fatty acids to form lipid polyesters. And also we could check that double mutant between the mutant overexpression of the transcription factor has very nice phenotype of resistance to accelerating aging, but when you cross with the loss of function of this enzyme, you lose completely the phenotype. Here, as I told you before, this HB25 is very, very conserved among angiosperms. So we transform with Arabidopsis transcription factor a species so distant as wheat and tomato. And as you can see, we got similar results. So very resistant seeds to accelerating aging methods, and also a reinforce of the seed coat. This we did in wheat and in tomato using also TEM microscopy. Finally, we did an experiment with the collaboration of Adriano with Pierre, and we got this publication. Seed coat lignification level is crucial in capsicum species seed longevity. Firstly, we did a mini screening with different varieties of Pierre with wild type accessions. We checked that very different behavior between the different varieties. In these varieties, in the most resistant and the most sensitive, we did some transcription analysis in the most important genes that were published in Arabidopsis, and this analysis, transcriptome analysis, allow us to determine that the varieties more resistant to accelerating usually accumulates more tocopherols, degrades faster the seed storage protein, as I showed you in this Kumasi. This is in dry seed. You can see that the protein profile is similar, but when you start the germination, these two accessions that are more resistant degrades the small albumins faster than the piquillo, that is, we can say, the wild type accession of variety. Finally, also we discover with microscopy and these several tinctions that all these varieties also have more accumulation of lignins in the seed coats. Finally, I wanted to introduce that what we are doing now. We did some metabolomics analysis, and we got some differential metabolic profiles between aged and fresh seeds. And choosing 3 or 4 metabolites, this is very nice because we can predict with high precision the germination of the seed that is going to be. And also we play with one of these metabolites. We order some mutants that reduce or increase the amount of this metabolite, and we'll see difference in the resistance to accelerating aging. Also, one of these metabolites influencing the expression of enzyme as so important as PARP3. Okay, I finished with to thanks all the work that we got with the collaborators, with Isabel, with Thomas Roch in Universitat of Innsbruck, and with Adriano Rodriguez in Comab. Also all my colleagues in the lab, and especially to Ramon Serrano that passed away very recently. So thank you so much, and yeah, questions you can do.
Thank you very much. It's outstanding, your work. So I think we have time for a couple of questions. I forgot to mention that those that are seeing the workshop through streaming, they can do their questions, they can write their questions in the comment section, the comment box.
Okay.
The people here in the audience, or the people watching us through Google Meet, if they want to, they can ask the question. Yes, go ahead, go ahead.
Hello, Eduardo. Thank you for your nice talk. I have a very difficult question for seed scientists. For your opinion, is there any correlation between the deep or dormancy and the longevity? Is there any correlation between dormancy and longevity?
Yeah, this is a very nice question, very treated in the literature. And we have, I think, we have two teams in this work. People, I think, is coming from Bensing, from Netherlands, that say it is, it's correlating dormancy as a resistant. I think, I don't remember well. I know that is the two teams, two opinions that say that if you are more dormant, you are more resistant to dormancy, and other teams that say the contrary, just the contrary. So less dormancy is more resistant. In my opinion, there are two different characters. So I think we have for AB25, I demonstrate, I have the results that is less dormant, but is resistant to accelerating aging. But I know, for example, I put in the slide that is all the, a lot of mutants in the ABA signaling, for example, the triple mutant SNRK, these are, they don't have any dormancy, but they have a lot, they are very sensitive to accelerating aging. For me, we have to this is a very important feature in the seed, but we have to treat differently. So some mutants are resistant and very less dormant, and there are more dormant mutants that are less resistant. So we have to separate the two features, in my opinion.
Yeah, I have the same. I think the molecular mechanism related to dormancy and longevity are quite different, so we have to treat that by a different way.
Okay, thank you for the question. Very, very nice question. We can discuss for ages with this.
Thank you very much. So could you please?
I have a question. If
okay, thank you.
Yes, so thank you for the wonderful presentation. I was curious about the fact that, prime the seeds, they show poor longevity. So I was wondering, so they are more susceptible to deterioration when they are stored in the long term. So I was wondering, or I would like to have a comment for you about the potential of your work, of your outputs in addressing also this kind of issue. I mean, you are gathering a lot of knowledge, relevant knowledge concerning this mechanism, and your mutants are also powerful tools for investigation. So, do you?
I don't know a lot of I don't know. I don't have a lot of knowledge about priming. This is the truth. I know that if you prime the seed, is very good for germination in short term, but you have problems because you humid the seed. This is not good for longevity, that is clear. And how can manage this? Maybe to find something, a treatment. I don't know. I'm very not related to priming, but I think it will be very good to get some priming or prime with some chemical, but maybe it's doing something to the seed coat. For me, longevity is very important. Seed coat is, for me, is the most important feature for seed coat. Or seed longevity. So I think if you prime with a chemical or something that reinforces the seed coat or impedes the entrance of the reactive oxygen species, should be good for longevity as well, for viability to long term.
Thank you.
But I think it's not easy, but yeah, it's the dual behavior of priming.
Thank you so much.
Okay.
Thank you very much. I think we can move to the next talk. Would you please, Alma, introduce the next speaker?
Yes, the next speaker is Adriano Griffo from University of Pavia. He's at the last, he has almost finished his PhD project, and he will talk about non-invasive methods to detect ROS as a proxy of seed quality. So, Adriano, the floor is yours.
So, can you see the presentation? Can you hear me? Do you see the presentation, the slides?
Yes, it's okay.
Okay, perfect. Thank you. So, good morning, everyone. Before starting, I would like to thank the organizer for inviting me to this beautiful event. As already mentioned by my first slide of the presentation, my line of research focused on the development of alternative approaches for the non-invasive estimation of seed quality. And as I will show during my presentation, we developed and tested different methods based on the detection of different markers of seed quality. So seed quality is the center of my research topic, and it is defined as the set of genetic, physiological, and physical features. And it is a very complex trait in terms of agricultural purpose, since it reflects the overall value of the seeds in terms of germination performances. So for instance, high quality seeds exhibit optimal germination performances in terms of speed of germination, synchrony of germination, and also percentage of germination, and are more resistant to different kind of stress, abiotic and biotic stress. So the utilization of high quality seeds can promote the improvement of crop production. And crop production, we all know that is probably one of the major tasks of modern society. We know that it must be increased to meet the zero hunger target of the United Nations Agenda for Sustainable Development. But the achievement of the zero hunger target is complicated by the climate change's effect, which are negatively involved into the crop production. So they induce the reduction, loss of crop yield, and also the steady world's population growth. So in this complex scenario, the utilization of high quality seeds may support the sustainable improvement of crop production and consequently the improvement of food production. So concerning the seed quality, an important aspect related to seed quality is that it's not an immutable feature, but it can change. So it can be improved, for instance, through priming technologies, but a natural process occurring in old seeds characterized by the loss of seed quality is seed deterioration. And the figure shown on the right illustrates a typical curve of seed deterioration. The time of this process can rely or depends from both intrinsic and extrinsic factors like storage conditions or intraspecific and genetic characteristics. But an interesting phase of this process is the first one, so the asymptomatic phase, in which we do not observe a loss of seed viability or seed quality, but this phase is immediately followed by a stage where we observe a rapid decay, a rapid loss of seed viability. And this trend suggests that some of the reactions or processes responsible for this rapid loss of seed viability are probably triggered or initiated, or probably are just occurring during the asymptomatic phase, but we cannot detect any change in terms of seed quality. Okay. So, given the importance of the utilization of high-quality seeds for agriculture, there are currently different methods available for the assessment of seed quality, divided into the invasive, which requires the direct consumption of seeds, and most of them are germination tests, tetragermination. So the most known and probably most used approaches for seed quality assessment, and most importantly, most of them are standardized by the ISTA, so the International Seed Testing Association. The non-invasive approaches represent a recent, I would say, alternative and more sustainable alternative to the invasive approaches. They are mainly optical technologies adapted and optimized recently for the estimation of seed quality. But despite the high number of methods available, no universal approach has been developed yet, since all of them present some limitation, like the time required for the analysis, the cost and the complexity related for the measurement of the analysis. So there is no universal approach for seed quality assessment. And in this context, it is collocated my line of research, which focus on the development of alternative approach, which are non-invasive, but also easy to use, rapid and cheap method for the estimation of seed quality. And during my research career, I work on the detection of different markers for the non-invasive estimation of seed quality, like the volatile organic compounds, the ultra-weak photon emission and delayed luminescence, which are two phenomena related with light emission, photons emission from intact seeds, and reactive oxygen species. And I will explore during the presentation each of every method regarding these markers. Okay. So one of my first activity focused on the development of ROS, the non-invasive detection of ROS. The present work was published with the following title shown illustrated in the picture on the left. So, as mentioned by Dr. Eduardo, ROS are controversial molecules with a dual nature. So they operate like single molecules and they control different processes. But when the overproduction or probably the overaccumulation of ROS occurs, they are involved into the induction of oxidative stress. So they are involved into the acceleration of seed aging process, inducing mitochondrial degradation, DNA and RNA damage, and so on. And in this context, one of the most important and most interesting theory or hypothesis justifying this coexistence, this dual role of ROS, is the oxidative window proposed by Lee, which describes an optimal range, so the so-called oxidative window of germination, that is an optimal range of ROS in which we observe only positive effects on germination, so in which ROLI promotes an optimal germination without inducing the negative effects, so the oxidative stress effects. So in this work, the idea was to estimate in a non-invasive manner ROS levels. So in the experimental system illustrated in this slide, as you can see, we worked with soybean, but we also worked with tomato and wheat seeds in order to measure ROS levels from high and low quality seeds we worked in a contrastive system made up of seeds treated with hydropriming to improve germination performances, so seed quality. And for each species, we selected 3 different priming protocols, but we also wanted to reduce the germination performances, so we use heat stress treatment, so we simply damaged at elevated temperature, 90 degrees, our seed material. Moreover, we measure the ROS level from the HB group, that is represented by seeds with just or only the inhibition phase of the hydropriming protocols. And the methods proposed for the non-invasive estimation of seed quality are the one illustrated in this figure. So the FOX1 assay and the DCFHD assay, which are 2 different biochemical methods that provides different information regarding ROS levels. So the first test used is the DCFHD assay based on this reagent, the fluorescein, that is able to react with different groups of ROS. In fact, this test provides a general overview, so a general measure of the oxidative stress level, measuring the fluorescence of the working solution after incubation period with our seed group. The second assay used is the FOX1 assay, which is specific for the detection of hydrogen peroxide and peroxidic radicals released from seeds, and it is based on the equation of formula that illustrated here. So these radicals promotes the oxidation of iron, and the oxidative form of iron can be detected with the xylene orange, that is an important reagent of the FOX1 working solution. And this complex can be detected with the spectrophotometer at 560 nanometers. So both the approaches were used for the non-invasive detection of ROS level. But before moving to the measurement of ROS levels, we wanted to validate our experimental system. So we wanted to confirm the fact that we were working with high quality and low quality seeds in our system. So we performed some germination tests with hydroprime and heat stress seeds for each species utilized in this work. And in the figure is shown the time course of the percentage of germination during all the time of the germination, plus the histograms showing the length of the root length, or in the case of weight and shoot lengths. So from the figure, we can immediately see the positive effect of hydropriming, so a positive improvement of the germination percentage, especially during the first day of germination, and a positive effect testified by the increase in root length compared to the control group, so to non-treated seeds. At the same time, we can also observe the negative effect of the heat stress on seed quality, as shown in the figure, so the low values of germination percentage as well as the low values of root length. So we validated our test, we confirmed that we had and we were measuring ROS from high and low quality seeds, so we moved to the measurement of ROS levels performed with the DCFHD assay in green histograms and in orange with the results of the FOX1 assay. So we can immediately observe that there is no general trend regarding the ROS levels or regarding the results of the fluorescein assay. So there is no general trend concerning the oxidative stress level, but we can observe a general trend in terms of hydrogen peroxide and peroxidic radical released from seeds in all the species tested. In particular, we can observe that control and heat stress group present higher values of ROS released compared to the prime and inhibited seeds. So from the results obtained, FOX1 may be tested or may be proposed with further validations as a potential alternative for the non-invasive detection of seed ROS, ROS measurements. And since we wanted to perform or wanted to develop a non-invasive assay, We also evaluated the possible toxic effect of this working solution to our seed. So we perform a germination test on seeds treated. So after an incubation time with the fluorescein with the working solution of the FOX1, and the results concerning the final germination, percentage of germination at the end of germination test is shown here in the histograms. And we can see that there is no significant differences in terms of final germination percentage among control and treated seeds. So based on the results obtained, the tests are not toxic or not toxic to our seed material. Finally, for this work, we perform also a principal component analysis integrating together germination and ROS data for each species. The scores plot show the clusterization of the different groups, different treatments on top of the slides. And here we can notice the clusterization of three major groups: the heat stress in magenta, so this one, the control group in red, and the hydroprime and inhibited groups, which clusterize, so they are overlapping, indicating a similar behavior in terms of germination and ROS levels. So the different priming protocols have an overlapping position, so they have probably a similar behavior in terms of ROS and germination data. But we can also observe with the B plot in the lower part of the presentation of the slide the contribute of the single types of data to this clusterization. And we can notice for each species that data coming from the ROS measurement, so from FOX1 and fluorescein assay, contributes to this clusterization. So confirming the results obtained from and showed in the previous slides. So from the results obtained in this topic, we may support the utilization of FOX1 as non-invasive approaches for seed quality assessment, but of course new experiments are going to be produced to further validate these results. Second topic I had the pleasure of being involved is the development of non-invasive seed quality assessment methods, detecting the ultraviolet photon emission and the delayed luminescence or fluorescence, which are two phenomena related with the photons emissions from seeds. And thanks to the Jeff Shell Fellowship for Agricultural Sciences, I had the pleasure and the honor of working at this topic at the IPK Institute of Gottesloben in Germany, using the seed material and the facilities of one of the biggest germplasm gene bank of the world. So just to introduce the two phenomena, the UPA, the ultraviolet photon emission, is the chemiluminescence released from biological systems like seeds, where electronically excited species are formed during metabolic and oxidative stress processes. So the releases of photon is induced by these species, electronically excited species, which in a complex mechanism, mostly unknown, they are able to dissipate part of their energy and induce the release of photons. The UPA can be spontaneous and induced, depending on the presence or not of external stimuli like light, high temperature, and also biotic stress. And the following figure illustrates the typical trend in terms of intensity, so frequency of photon release over time, of a spontaneous UPA in blue. So we can notice that this trend is constant over time, so the intensity is constant. And also an induced UPA trend, which is quite different because we can observe two stages. A first one when we have a high release of photons, followed by a rapid decay, and a second stage in which the intensity, so the release of photons, is constant over time, where the first stage of an induced UPA is the delayed luminescence phenomenon. And we develop a method to use this phenomena as hallmark for seed quality estimation, because UPA and DL are linked to the physiological state of seeds. So they are very informative phenomena of seed quality. And this is quite logic because we knew that UPA, as already mentioned, is related to the oxidative reactions. So the frequency of occurrence of oxidative reactions has an effect because it induced a loss of seed quality, but it also changed the natural pattern of photon release. Moreover, for delayed luminescence, different works highlighted a correlation between this phenomenon with some features connected with seed quality, like seed viability, water content, rust production, but also chemical composition. So they are very informative phenomena of seed quality. And in the present work, which is in submission, the aim was the measurement of this phenomena from seeds of different species, so different seed group of 5 different legume species. These seed groups were also used to perform germination tests, and these two types of data, the UPNDL measurement and the germination data, were integrated together, and the final datasets obtained with this integration were used to develop machine learning models able to predict the outcome, so in our case the class, so the percentage of germination, based on the measurement of UPNDL. So, as indicated also in the experimental design, we work with 5 legume species, different accession for each species, and an interesting aspect of this work, which was possible thanks to the great facilities of the IPK Gene Bank, is that for each accession, we could measure the UPNDL from an ambient group, so naturally aged seeds, so seeds stored for more than 10 years at room condition, and seeds belonging to the cold group, so seeds stored for more than 10 years at -18, which is probably one of the best available storage methods to better preserve seed quality. The results obtained are very encouraging because we performed the validation of our predictive models, performing cross-validations, and the accuracies obtained with this validation are quite, and in some cases, in some species, very positive, which highlight a correlation between this phenomena and seed quality, and also the possibility of using these phenomena as indicators of seed quality. Another recent project I had the pleasure to work in collaboration with the CNR EPSP Institute of Napoli Portici, thanks to a fellowship, so the support of the Italian Society of Plant Biology, is the non-invasive and real-time detection of volatile organic compounds using a specific technology that is the proton transfer reaction time-of-flight mass spectrometry, which is often associated to gas chromatography mass spectrometry for this kind of analysis. So VOC are very informative, molecules very informative of the state quality, so the status of seeds, because they represent the byproduct of many metabolic reactions. So their emission from seeds provide information regarding the occurrence of specific reactions, like lipid peroxidation, which is an important process related to the loss of seed quality. So they are very informative molecules. So in the experimental system of this work, which is still in preparation, we work with different species, and 3 of them are orphan legumes included in the Benefit Med project. So we also detected the volatilome with these technologies of 3 orphan legumes. And we also, in this case, in order to perform this kind of analysis in a contrastive system with high and low quality seeds, we treated our seed material with hydropriming to improve germination performances, and with artificial aging protocols to induce the loss of seed quality. And then we perform the analysis with the PTR, TOF, MS, and we perform data processing. And for now, the data elaborated so far are quite interesting because they illustrate a trend in which there are clusters of volatile organic compounds differently emitted among treatments. So there is a difference in terms of volatilome between hydroprimed, controlled, and artificially aged seeds, but also among the different accession of our seeds and among species, which suggest an intrinsic differences which can, in terms of metabolic activity, which can be support or which can confirm the different release of VOC. So there are also differences in terms of the volatilome among species and accession of the same species. So I concluding my presentation, and I just want to take the opportunity of thank all of you for giving me this beautiful chance, my supervisor, Professoressa Magovei and Professoressa Balestrazzi, and all the research and expert I had the pleasure to work with, Professor Borner and the team of the Italian Institute of Portici, the CNR EPSP. So thank you for your attention.
Thank you very much. Very nice. Could you please thank you very much. Thank you for your talk. It's very nice. Great deal of work and interesting research lines. I think we have time for a question. Here in the hall, do we have a question about what Adriano? Yes, Loic has a question.
I have a short question. For the non-invasive method for ROS detection, is it easily adaptable for different species?
Now we tested on soybean, wheat, and tomato. So since we observed, especially for the FOX1 results, a general trend we think it's possible to use for other species, of course. Probably for each species, it occurs some adaptation. So for instance, the time of incubation of solution must be adapted. The number of seeds in case we want to perform bulk seeds, but yeah, I think it can be adapted. It's our purpose after all, so to develop methods that can be adapted and used in all species. So our results collected so far suggest that, yeah, it will be possible.
Okay, thank you. Congratulations for this work.
Thank you very much.
Well, thank you very much. I think we are kind of running a little bit, so I think we should move to the next talk. Could you please introduce, Alma, your next speaker?
Yes. So I'm very happy to introduce Loic Rajjou from Institute Pierre Bourgin, France, Agro-Paritech, Paris-Saclay University. It's a cluster of institutions. So we all know his seminal work on seed biology. So please, Loic, thank you for being here, and the floor is yours.
Sorry, I have a problem because I'm not allowed to share my screen. I don't know why. Sorry, I changed my navigator because I was not able to share my screen. So, want to share. Open.
Have a square just underneath your screen. You'll have like a square with an arrow. Well, left.
Okay, now it should work. Okay, did you see something?
No.
No.
I just click on the presentation so we can see it in enlarged.
Is it okay?
Yes, now, perfect, thank you.
Okay, so thank you very much for inviting me. So I will give a talk. My title is The Benefits of Seed Enhancement through Chemical and Biological Priming and Seed Priming. So my name is Loic Rajjou. I'm working in Versailles in France in Institute Jean-Pierre Bourgin. It's a joint unit between INRAE, AgroParisTech, and Paris-Saclay University. Just to remind the concept of seed quality, when we talk about seed quality in agriculture, we talk about physical quality, we talk about genetic quality and specific purity. We are talking about physiological quality associated to germination capacity, and the last point is the concept of health quality in order to have seeds without any pathogen or pest. And in order to test seed quality, we have to follow the recommendation of ISTA, which is an international association that with a different kind of recommendation in order to evaluate the quality of seed lots in agriculture. In order to protect seed or to stimulate seeds, there is a different emergent pathway, and notably what we call the bioprotection, meaning using natural-based substances or biological control agents like microorganisms in order to protect plants. And we can apply this concept of bioprotection to seeds in order to improve quality. It could be the improving of physiological quality and stimulate germination with natural substance or microorganisms, and also to protect seeds against pests and pathogen using biocontrol agent or stimulant of defense mechanism. So if we are thinking about the seed for tomorrow, we have to integrate the idea where we are in ecological transition. We have more and more food and technological used, and we have a cycle of bioeconomy in agriculture. And in this way, so securing plant production, meaning securing seed quality, is very important for all the agricultural chain value, and notably to produce animal feed, to produce food for human in terms of quality and quantity, but more and more also to produce the bio-based materials, bioenergy, and also to produce high-value compounds for agriculture, health, or well-being, like specialized metabolites, alkaloids, terpenes, flavonoids, etc. We have to take in consideration the genetic and genomic quality of plants, of course. We have to take in consideration the climate variation. It's a variable. The policies and evolution of policies, notably in Europe, and the demography and social behavioral trends. So, in order to have good crops and very performant agriculture, like on this picture, and in order to avoid bad efficiency in yield, we have to take in consideration all the social, environmental, and regulatory demands. And this happens today in new markets and new differential products. In order to address the Green Deal, the European Green Deal requirements towards a synthetic pesticide-free agriculture in Europe. So there is different way to address these requirements, and we have different transition in terms, for example, in plant breeding with different novel technology and changing in the policy about gene editing, GMO, novel gene technology modification, the use of synthetic chemicals, which are more and more banned in Europe, an increasing in the use of chemical from organic chemicals, how to use microorganisms in agriculture. So it's a very important question. And there is a couple of other technology associated to regulatory European system in order to address these issues. So to do that and to deal with that, we need to solve problem, and to solve problem, we need to make science. And to address the question of seed priming is a very old technology, consisting to deal with water potential in order to imbibe seeds, okay, but also to prevent germination before drying seeds in order to get a prime seed. So to do that, we need to control the water potential by controlling the matrix potential, the osmotic potential, and the pressure potential in order to prevent testa rupture and radical emergence and drying the seeds. So prime seeds generally germinate more rapidly, as you can see here on the red curve, than untreated seeds. And the germination is more uniform. So what we expect from seed priming is usually applied to commercial seed lots. It's widely used by seed technologists in the seed industry and seed company to enhance seed vigor and seed quality in terms of germination potential, and also to increase stress tolerance. But in most cases, it's to deal with abiotic stress and not really to deal with biotic stress. And so to meet these requirements, we probably need to do more science to understand mechanisms associated to seed priming to develop novel methodology. So if you can see this curve, here you have a germination rate and the distribution of germination over the time. When you have prime seeds, the seed lot germinate faster, as you can see here. The germination is more uniform than the unprimed seeds. And when you look this other picture, the prime seeds are able to germinate under a wider range of temperature, under cold temperature or elevated temperature. And the priming is also able to eliminate a dormancy factor in order to improve the amount of germination slightly. So we expect to have a better germination. So it's a technology which is easily scalable. You can do a scale up from lab to industry. There is different equipment existing on the market to do a priming with Durham, with osmopriming system, etc. And what we expect, there is different way of priming. You can use chemical priming using different synthetic or biological chemicals or extract to add in the water. You can use nanomaterials or nanoemulsions in the water, and you also can use biological priming using microorganisms or natural products inside the water. And what to expect is if you have a non-prime crops pathogens with a high pathogen susceptibility, we expect to increase the plant biomass and the resistance to abiotic stresses But now we also expect an improving in disease resistance. But to do that, what we know is priming will change a lot of physiological parameters at the scale of the seeds and the cell inside the embryo. You will change the reactive oxygen species and burst, and notably the Ross homeostasis. You will change the transcriptional activities with transcriptomic reprogramming. You will change metabolomic activities inside the embryo, and something which is not widely studied, but we can expect to have epigenetic modification, like methylation or histone modification, in order to have a kind of memories along all the cycle from seeds to seeds during a crop production. So what we expect as a benefits, in order to address this question, we can have different kind of focus. We need to improve our understanding in order the enhancement of seed defense by priming system. There is a lot of black box which we are not really understand what's happened in terms of seed defense in order to cope with biotic stresses. We also need a second focus to increase our knowledge about the crosstalk between seeds and environmental microbiota in the soil and directly associated to the seeds. And if we have both, we can address another focus is the impact of biocontrol strategy for seeds in order to connect biocontrol and seeds to propose novel strategy of seed priming. So there is different research issue and working hypotheses. What we know is the perception of their environmental by plant lead to the implementation of cellular and molecular adaptation mechanisms to cope with situation of constraint, biotic or abiotic. So currently, the current state of knowledge regarding the defense and adaptation system of seeds and growing seedlings remain poorly described by comparison to leaves or to roots. So we need more effort in terms of science, and to characterize the metabolic and molecular defense mechanisms of seeds through their development, maturation, germination, and including storage, in order to consider a relevant strategy for enhancing this process in association with biocontrol solution, including microorganisms. So I will show you two examples of work we perform in my lab. The first work is dedicated to understand better the defense mechanism related to seed priming with hormones involved in plant defense. My example focus on salicylic acids. I previously worked on salicylic acids. Maybe you know these molecules. It's a samples molecules. It's a plant hormone involved in a defense mechanism in plants in order to deal with biotic pressure by a pathogen or by pest. If you applied salicylic acids in the water during seed priming, here you have a germination after 36 hours after imbibition. You have a non-prime seed lot, a prime seed lot, so you improve the speed of germination, okay? And if you use salicylic acid to 500 micromolar, you can improve significantly the rate and the uniformity of germination. As you can see on the picture, you have seedlings very uniform in my Petri box. And in addition, if we measure 15 days after sowing the dumping off in presence of a pathogen, here we use a pathosystem with Arabidopsis and a fungi called Alternaria. We infected the seeds with Alternaria, and we infected unprimed seeds, primed with salicylic acid seeds. And the prime seeds remain very sensitive to the attack of Alternaria, and we can measure dumping off. And when we applied salicylic acids to 500 micromolar, we can reduce the impact of the pathogen and reduce the dumping off. So it's a good news, because by seed priming with phytohormones involved in plant defense, we are able to improve the resistance to pathogen in the seedlings. So in order to understand what's happened, we performed some transcriptomic work by transcriptome analysis of prime and unprimed seeds with prime and with salicylic acids and prime with water. When you prime here we have 6 samples, and we can see on this PCA there is a very clear transcriptional word programming, because we can separate easily on the first component the samples prime with salicylic acid versus the samples unprimed, prime without salicylic acid. So we look for differential expressed genes, and we found 271 up-regulated genes and 184 down-regulated genes when we compare prime seeds with water and prime seeds with salicylic acid. There is some genes which are more important in terms of differential expression, and I will show you just a few examples. We expected to have only 2 clusters, genes which are up and down regulated. When we perform a clustering quality descriptor, as you can see on this heat map, actually we don't have only 2 clusters, but we have 5 clusters of genes with correlated expression, and we can deal with that, and in order to understand which genes are co-expressed or with up and down regulation. So we perform some gene networking, gene regulatory networking, with all the genes differentially expressed up and down. We use a tool called Geni3, and we can identify by this way 12 master regulators in 5 main clusters that responds to salicylic acid in order to modulate the gene expression probably related to gene defense. So just show you two examples. The main example is this protein, which is a transcriptional coactivator. The expression of this protein of this gene was described as a specifically elevated in response to pathogen infection, to salinity, to drought, to heat, but also to abscisic acid or salicylic acid. So it was a good markers. And there is another gene involved in its RNA helicase family protein, which is clearly downregulated in seeds after salicylic acid treatment. We look for markers of salicylic acid action, like NPR1. NPR1 is a transcriptional factor induced by salicylic acid, and we didn't observe variation in terms of the expression of NPR1. But when we look at other genes involved in the response of salicylic acids, we found different genes involved, like NPR3, NPR4, which are receptors for the immune signal of salicylic acid. And it depends of the concentration of salicylic acid. According to the concentration, you don't have exactly the same response in terms of susceptibility or resistance to the pathogen. So NPR3 as well NPR4 are control NPR1 homeostasis. So NPR1 is the main markers of salicylic acid response, and in concentration dependent way. And when you have very high concentration of salicylic acid, you can induce an interaction between salicylic acid and NPR3 that interact with NPR1 to have what we call effector triggered immunity, ETI. So ETI at very high salicylic acid concentration is appropriate to attach to NPR3 and stimulate this association to NPR1 and resulting in NPR1 turnover. And in this case of ETI, there is something that is very interesting. There is no antagonism between salicylic acid and another hormone involved in defense called jasmonic acid. And that means that probably during the early step of germination, these two hormones are able to discuss all together without antagonism. So there is a described model in literature describing that. If you add salicylic acid in low concentration, you will have a transient response in terms of resistance or vulnerability in response to mechanism involved in plant resistance. If you have an ETI response, you can have a prolonged compensatory response and a robust response to deal with pathogen. And it's what we observe with the priming of seeds with high concentration of salicylic acid. So this is a good news because we can expect to have improve seed vigor by improving germination uniformity, by improving the speed of germination, but also by improving the response to stress, including both biotic and abiotic stress. The second aspect of my talk is to consider the seed microbiota. In the field of seed industry, plant breeding people like to say we need to have good genetics and we need to have good seed lots. And there is an emerging idea saying that we also need to have a good microbiota in order to perform good germination performance in the field. So what we know, germinating seeds interact with their biotic and abiotic environments, and they probably have some co-evolution between seeds and their beneficial biotic environment influenced by abiotic factors. So what we know now that plants and seeds are associated with many microorganisms, and most of them are non-pathogenic and can promote some physiological aspects of the plant. But what we know, it is very well described in this paper published last year. Seeds correspond to a bottleneck in terms of diversity and biomass of microorganisms by comparison to other plant organs, by a reduction of the microbial inherence in the plant cycle. The interaction with environmental microorganisms include for seeds associated with different kind of microorganisms like fungi, like bacteria, but also like virus, etc. And when you imbibe seeds, you can shape the microbiota in order to shape the future microbiota associated to the phyllosphere and to the rhizosphere in order to be in competition with potential pathogen in the soil. So there is another paper describing what we can expect from the good association with seed microbiota. They published very different work published describing that according to the plant group, you can find different fungal taxa and different bacterial taxa, and in order to deal with different kind of pathogen. So it's very interesting to focus on that, because focus on seed microbiome can help to optimize different benefits, notably in terms of nutrient uptake, notably in terms of protection to develop antagonism of competition mechanism with disease and pathogen, to improve the quality of soil and quality of water by microorganism and improve phytoremediation mechanism, to stimulate the plant metabolism by producing different molecules by the microbiota, like phytohormones or ROS synthesis, and to deal with abiotic stress when microbiota is associated with the seeds. So in order to address this question, in my lab, we decided to explore more deeply the spermosphere. Spermosphere is a zone just around the imbibed seeds of biological influence, which is described previously by an Italian guy, Verona. The spermosphere is an important interface between seed and microorganism in the soil. It's the main energy source for seed-associated microorganism is the carbon released by seeds. And we know now seeds are able to release RNA, proteins, carbohydrate, peptide, and metabolite in the spermosphere in order to feed microorganisms, beneficial but also pathogen. And spermosphere has an only temporary existence during the germination before radical emergence and the installation of the seedlings. So to explore that, we use a model. We use Phaseolus vulgaris, common bean genotypes, and we explore the spermosphere by different ways, describing microbial community dynamics during germination at different time of kinetics, describing metabolite, peptide, and RNA released by the seeds, and the crosstalk between the seed component released in the spermosphere and the dynamics of microbiota during the time course of germination. I will just show you one result. It's a network describing the interaction and positive and negative correlation between metabolites released by the seeds and the bacteria and the fungi in the different genotype produced under different contexts of environment. And what we observed, there is some metabolites which are clearly correlate or anti-correlate with the presence of different strain of microorganisms. Just an example of a very interesting bacteria called Bacillus megatherium. Bacillus is already used in soil amendment in agriculture. And we can observe that there is a negative correlation between the presence of Bacillus megatherium in seed spermosphere and the presence of flavonoids. And in contrast, there is a positive correlation between the presence of Bacillus megatherium in the spermosphere and in the presence of amino acids and derivatives, meaning small peptide, dipeptide, tripeptide. So this is interesting because there's meaning we can use Bacillus megatherium in biological priming solution, but the condition is to add the good metabolites in order to promote the development of Bacillus megatherium in the seed priming technology. So the take-home message is the seed seems to be able to shape its own microbiota during germination by releasing a high diversity of molecules in the spermosphere. The composition of the spermosphere is very dynamic and selective for the microbiota, so it could be a good source of idea to select a good microbiota and to adapt this microbiota to seed priming environment. And the spermosphere looks to be a relevant interface for characterization of natural substance for seed and plant protection against pathogens. So I will conclude. In order to adapt to seed priming, we will probably to develop interface also with chemistry of nanoparticles, because nanoparticles could be a very promising carriers for delivery, metabolite, peptide, microRNA, or microorganisms to living systems, including in seeds. And we can expect to develop in the seed priming context novel kind of nanoparticles in order to carry biostimulants, hormones, and even microorganisms, and adapt to other kind of seed technology like coating and pelting, and to combine all of this in order to improve the quality of the seed. So in my mind, the future prospect and new way for chemical and biological seed priming, it's maybe to think out the box and to focus on biocontrol and to focus on the identification of novel bio-based molecules or family of molecules of extract, to focus also on microorganism and microorganism communities, and to adapt that to formulation in order to develop novel kind of priming and seed treatment like pelting and coating. We could also combine seed priming to other novel technology developing in seed technology, like the application of cold plasma in order to develop disinfection without any chemicals. So which could be very interesting. And cold plasma also affect the tegument and improve permeability and disequilibrate the balance of reactive and oxygen species and nitrogen species. And maybe we have to focus on that to combine seed priming treatment after a cold plasma treatment. And of course, we will need to develop knowledge in terms of seed biology and adapting maybe modeling techniques, either statistical modeling, but also deterministic modeling, in order to have a good view about metabolic fluxes and using a model like FBA or FBA models, in order to adapt the genotypes of the seeds with the microorganism and to predict or to design a good microorganisms communities adapted to a good genotype. And to do that, maybe in the future, it's not today, but artificial intelligence, and notably protein language model and large language model, can help to design the good genotype for microorganisms and for the plants, and to adapt all of these in order to develop the good treatment. So it was my message. So we need science to develop very efficient and very smart priming in the future. So thank you for your attention.
Thank you very much. A very insightful and very exhaustive presentation, highlighting, I think, the most important points in the mechanisms of seed priming. Unfortunately, we have now to take a break, and we don't have time for questions.
Okay.
So thank you very much. We will resume the workshop at 11:30, I think. So if people watching us through YouTube, they want to write some questions, please do, and we'll transfer those questions to the speakers on due time. Thank you very much. Hello, we will move soon.
Hi, we are here.
I know you can see it, but we got more audience now, students.
Good, good.
So they had lectures this morning, but now they were able to come.
Good.
Welcome to the second term of the workshop. We will move to the next talk by Professor Beatriz Julián from the Institute of New Advanced Materials from the Universitat Jaume that will present some insights on new biocompatible materials for sustainable agriculture. Dr. Julián, the floor is yours.
Thank you very much. Just let me check if the presentation is here. Yes. Can I share here? This. Is it okay? How can I put the whole screen, the full screen? How can I put the full screen? Sorry.
You move from here, actually.
Okay. So, first of all, thank you very much. I would like to thank Vicent Arbona and his colleagues that are organizing this workshop. It's a pleasure to be here today and welcome also some students from our Master in Chemistry here in Castellón and all of the people that are attending online. I would like to apologize, first of all, because I'm not from this field of agriculture, so I'm a chemist, inorganic chemist indeed, and I usually work on materials for different applications. As you can see here in this picture, you can see several suspensions of nanoparticles that are fluorescent. They are semiconductors, and we usually prepare these nanocrystals for LIDs, so for lighting devices. So this is not, as you can see, this is not my field of expertise today, but I will try to, I have been learned a lot about the use of nanoparticles and nanomaterials for agriculture, sustainable agriculture, and I would like to share with you some of I would like to give a perspective and some critical perspective for those who want to work in this field. So how can we go from these nanoscale materials to to more sustainable agriculture? Well, this is the outline of my talk. I will start with a brief introduction, and I will just give an overview of some applications of nanotechnology in agriculture, the type of nanoparticles that we can use with, the synthetic methods and internalization processes in the plants, the role of nanoparticles in crop protection and plant growth, and finally, I will give some conclusions. Well, this is the main goal in our era. We have two major challenges in this century. We need to get zero hunger, and also we have a climate action. This is our challenges, and In particular, climate change is significantly impacting on agriculture through direct and indirect effects on crops, soils, livestocks, and pests. So how this climate change is affecting the agriculture? So it alters the weather patterns, increases temperatures, changes precipitation levels, reduce crop yields, shift planting seasons. This is the case of wine. Do you know that wine, typically in Spain and France, all the vineyards are moving to United Kingdom and more places in the north of Europe, and also increases droughts and floods. So this impacts a lot on agriculture because as you can see here, there is a loss of the overall crop yield. There are alterations in nutrient dynamics, decreasing photosynthesis, and so many effects that the climate change is affecting due to the global warming, the extreme weather, and also the rainfall intensity. So in the past decades, agriculture technologies have been successfully in eradicating hunger from many parts of the world, but by using chemical fertilizers. And this has really been very successful, but the excessive use of these fertilizers that provide nutrients to plants like nitrogen, phosphorus, potassium, etc., has increased the agriculture productivity worldwide, but these fertilizers have caused more harm than good in long-term perspectives. So therefore, the modern agriculture sector needs more clean and green strategies for simultaneously improving crop productivity and mitigating climate change impacts. So here is the nanotechnology can help us to get more sustainable agriculture, and in particular the use of nanoparticles can affect or can be useful in different processes for agriculture. For instance, we can increase productivity, for instance using nanopesticides or nanofertilizers. In particular, we can think on different inorganic nanoparticles, but also polymers, or even lipidic nanoparticles. We can also improve the quality of the soil. Take care of the soil is very important, as we have already seen in the previous talks today. For instance, the use of nanoclays or nanozeolites or hydrogels can help to keep the water in the soil, for instance, and also to introduce or incorporate some nutrients, etc. Also, as today we have also seen the priming seeds is helpful to stimulate the plant growth. And in this case, a lot of nanoparticles have been used, for instance, gold, silica, titania, zinc oxide, also carbon species like nanotubes, fullerenes, etc. And in the end, also we can find very helpful and useful nanosensors that provide smart monitoring of the situation of the plants and our crops. This is another image, a little bit more complete, but I wanted to show it to you how nanotechnology is helping in agriculture. It's more or less the same, but I would like to show you here the possibility of using nanoparticles as nanocarriers, for instance, silica or chitosan or lipids, etc., or clays that can be loaded with different active species that can be insecticides, fungicides, herbicides, etc. Or even we can modulate or incorporate some RNA pieces, fragments. Well, let's move. Yeah, so these nanoparticles can deliver agrochemicals. And these agrochemicals can be peptides, nucleic acids, small molecules, through different kind of nanomaterials, as I already commented, silica, polymers, metallic nanoparticles, etc. These nanomaterials can be used as raw materials or with or without surface modification in order to target specifically a part of the plant. and also to provide these nutrients to the specific part. And this can be very helpful from an economical point of view. Nanoparticles charged or loaded with different species can also be useful to cure plant diseases and to improve nutrient levels, fertility, and soil health. Here you can see there's different effects. Here is the bringing the target, are for instance the raven to the leaves, or here we have a activity for to cure these plant diseases, or here a lot of different carbon species, nanotubes, fullerenes, lipidic species, vesicles of different organic molecules charged with different species. Well, the type of nanoparticles is very, very extensive, has been extensively explored, a lot of nanoparticles, not only in nature, but also in morphology. Morphology is very, very interesting because we can work with 0D or 1D structures, 2D and also 3D. But all this morphology will affect drastically to the activity of the system. But also the nature of nanoparticles can be organic or inorganic. In organic, we can select or work with carbon nanoparticles, polymersomes, dendrimers, etc. And from an inorganic point of view, we can use directly metal nanoparticles like gold, silver, copper, etc. And metal oxides like silica, titania, zinc oxide, etc. And also dendrimers, inorganic dendrimers. So, as you can see, the panorama is really, really very extensive. How can we prepare these nanoparticles? Well, usually when we want to go to the industry, some people use the top-down methodologies, meaning that from a bulk material we start to make fragments, and in the end we reach the nanoscale structures. But this is really, really complicated because the system, the milling, laser ablation or sputtering is quite complicated to get monodisperse in size and in nature nanoparticles. But I would say that the most useful is the bottom-up approach, in which we start with molecular species or even ionic species that start to react and to enlarge, to make some clusters, and in the end nanoparticles or vesicles, etc. I would say also organic molecules can make self-assembly and get higher, more complex structures. Inside these structures we can embed different species that are of benefit for agriculture. Well, I was surprised that some people in the field are trying to do green synthesis by using plants and microorganisms like bacteria, fungi, yeast, or viruses to generate nanoparticles inside. This is a tendency that is quite accepted, or at least this is what I have, this is the conclusion I have obtained from looking at the literature. This is quite appreciated as a quick, facile nanoparticle mass production of stable and biocompatible nanoparticles. And specifically, it seems that some medicinal plants are reported for the rapid synthesis of metal nanoparticles like gold or silver. For instance, the synthesis can be, these particles can be obtained or synthesized and grow in few seconds to few hours, like bacteria synthesis, which often requires more time, like 1 or 2 days. And you can see here, this could be the green nanoparticles that are, I would say, more biocompatible because they have a biological corona that is, of course, of importance for the final applications. Okay, so as we mentioned, the corona or the surface properties of nanoparticles are absolutely crucial because this is the surface of the nanoparticle that will be interacting with the plants, with our system, with the cells. So in this case, I chosen a figure from this paper, and it's a core. This is the core of a gold nanoparticle. Gold nanoparticles are very useful, very used in different fields, in medicine, biology, chemistry, physics, electronics, cosmetics, a lot of fields of interest for gold nanoparticles. But specifically in agriculture, it seems that these nanoparticles work best at lower concentrations and improve physiological parameters under normal and abiotic stress conditions. But of course, when the nanoparticles enter into the plant, some conditions will change. And for instance, these particles, the aggregation, we want to avoid aggregation of nanoparticles, and this is usually very influenced by the ionic strength of the media. So in case of high ionic strength, the nanoparticles will tend to agglomerate, and this is an effect that we don't want. So the easiest way to control this agglomeration is by trying to put some spherical effects or charge effect at the surface, try to avoid this attraction and get repulsion in between the nanoparticles. And usually some organic molecules can be are used to make this to create this corona. So from small organic molecules, amino acids, proteins, antibodies, polymers, or charts, as I said, or even surfactants that has some specific properties of being amphiphilic, meaning that they have a polar part and an apolar part, and they arrange, or they organize their polar head or the apolar tail in the sense that the interactions are better, okay? So once the nanoparticle with the shell enters into the system, we can also trigger or modulate the hydrophilicity or hydrophobicity of the nanoparticles, and this will help to make stable suspensions and dissolutions that will come and pass through the plant and the cell membranes. Okay, so the general modes of uptaking the nanoparticles by plants, taking into account that usually the plasma membrane has a phospholipid bilayer with hydrophilic heads and hydrophobic tails, as I mentioned. There are like three main processes to uptake these nanoparticles. The direct diffusion processes, the endocytosis, and the assisted and uptake assisted by transmembrane proteins or channels that regulate the movement of the nanoparticles into the cells. And also this uptake will be modulated by the way of application of nanoparticles in our plant, because we can, for instance, put or apply nanoparticles in the root. So from the root system through the xylem to the shoot, and no downward movement occurs. Or we can spray, make a foliar spray of nanoparticles, and the way of application of the application of these nanoparticles will affect their activity. Here I found very interesting this image where you can see different type of compositions of nanoparticles. Here you can see some laminar solids like nickel hydroxide, iron oxide nanoparticles that are well known in treatments for hyperthermia. These nanoparticles are very useful in bio systems, because you can apply not only a magnetic field, and the system will start to heat. The nanoparticles start to heat. But also cerium oxide, titania, zinc oxide, which is well-known semiconductor, carbon nanotubes, fullerenes, these nanoparticles, gold, silver, copper, etc. So I thought this is quite interesting, and how these particles are distributing in the different parts of the plant. If you can, you want to. I don't have time to explain a lot, but just to have a general idea of how we can trigger, depending on the material, you can target one specific part of the plant or other. And this, you can see how these nanoparticles can enter into the cellular system by different processes. I don't have time to extend, and it's not my expertise. But if you want to have more information, you have here the reference. Yeah. So how can we use these nanoparticles in agriculture? So the first use is in crop protection, of course, and we can use them as nanofertilizer. So we can charge our nanoparticles with some specific active species and release in a controlled way these active ingredients. So this is a more efficient delivery of active ions than the traditional way. And this, the good point is that results in lower amounts of pesticides and increased safety for humans. The regulated distribution of our ingredients minimize unanticipated losses in the form of evaporation in the plant, leaching, lixiviation, and other non-desired effects. Of course, we need important qualities in our system, complex system. We have to include the ingredient form. We have to keep it to make a good retention. We have to control the movement, and also the internal form of our nanoparticles, nanocarriers, to make a good delivery in the system that we want to work with. So these nanofertilizers or transporters lead to higher crop production and healthier vegetation. And so the macronutrients and micronutrients are encapsulated within nanoparticles and can be supplied directly in the form of nanoemulsions. Here are some examples of nanofertilizers delivering controlled release of nutrients for foliar and soil applications are hydroxyapatites, nanoclays, mesoporosilica, zeolites, chitosan, and metalloxides, as we previously mentioned. As well as nanofertilizers, also we can work with, or we can produce nanopesticides in order to control the weed, weed control strategies and the pests. These systems, in the nanopesticide system, we can include nanoinsecticides, nanofungicides, nanobacteriocides that are employed for their efficient delivery methods. Is cheap cost and easy of administration. And again, we need to encapsulate the active species in the nanomaterials. And the most useful nanopesticides will be those that are biodegradable, and for that we require many qualities. We have to control very well the regulated and delayed release, increase stability, the permeability within the target, and resistance to early deterioration. And in this case, as nanopesticides, some inorganic nanoparticles have been studied, as I said, as insecticidal, antimicrobial, and antibacterial against plant pathogens, aiding in crop loss prevention. And also herbicides, such as these species that are here, are encapsulated into polymers like alginate or chitosan. These are organic polymers, and have demonstrated significant efficacy against the target weeds. This is just one table where you can see the nature of different nanofertilizers, nanopesticides, and even bio-inspired nanomaterials, their activities, and what are the advantages of using these nanosystems. Also, nanobiosensors I think this is very important too, because this will help to detect the problems in the soil, the problems in the issues in the plant, by using some component that is biosensing. It introduces this biosensing behavior into a chemical or physical signal, an optical signal. And this signal, usually visual, electrical, or physical, can be acquired even at extremely low concentrations of the analyte or the parameter of interest. And here, I would say that most of the nanobiosensors that have been explored in the literature are based on gold nanoparticles, because they are very useful to detect the availability of nitrogen nutrients in soil or water, the moisture content in soil and its humidity, the content of carbon, phosphorus, and other elements, residual pesticides or poisons, for instance, or microbial infections, and also pollution monitoring. I think this is very interesting. And finally, for crop protection, we can think on nanoparticles for environment stability and nano-remediation. And I think this, the nanoscale materials are very useful in abiotic and biotic techniques to remove contaminants, but because these techniques have several advantages: the reduction in cleanup time, the elimination of complicated procedures, a reduction of cleaning cost, decrease in the intensity from certain pollutants to 0 levels, so it's good. And the methods for acting, the way of action is by absorption of the pollutants, or degradation, photocatalytic degradation in some cases, or by oxidation or reduction processes. So in this case, for environmental stability, the preferred systems are zeolites, iron nanoparticles, iron oxide nanoparticles. Here you have some of them, nanohydrogels, carbon nanotubes, etc. And these have been used for remediation of soil contaminants, target delivery of nutrients, and augment physical condition of soil. And also graphene-based materials are very, very useful because they have very large surface area, improve active sites, functional groups in its surfaces, and excellent chemical stability. And also, nanomaterials can be very useful on plant to stimulate the plant growth and development. Nanoparticles affect the germination of the crops. Here you can see one tomato plant, and the action of these nanomaterials can increase the metabolic and enzymatic activities, can reduce nutrient leaching, improve seed germination and root growth, enhance the crop yield and quality, etc. So I think the application of nanoparticles is very interesting. Not only the seed germination, as Professor Loic Rajjou mentioned, but also, oops, it's okay, yeah. But also in seed germination index, seed vigor index, and other parameters. Here, for instance, you have the silica, the interaction or the use of silica to, they studied the effect in growth of the, this is nano-priming using silica nanoparticles, and the effect in the seed germination. Can you see how, in comparison to the control, by adding different concentrations of silica, the growth of the small plant is increased. Yeah, I don't want to be very exhaustive, but just to put some, to highlight some points that I think to be critical. I think the use of nanoparticles is not easy at all, and it depends a lot on the concentration, on the mode of application, and the types of solids, and the quality of the air and the water of the system what we are working with. So for instance, depending on the type of nanoparticles, here you have some examples, you can see and observe a positive or a negative effect depending on the concentration. The same for the mode of application. In some cases, some nanoparticles, when they are applied in one way or another, the system is less effective. Or, as I said, it depends also the type of soil. For instance, in clay soils have negative influence on seed germination compared to sandy soil in some cases. So in the end, as always in our life, everything depends on the dose that we are using. For instance, gold nanoparticles, silver nanoparticles, zinc oxide nanoparticles, copper, depending on the amount, the dose that we are using and the size, it can be safe methods, they can be a safe method, or we can found the material is toxic for the plant. So the system is quite complex. Using nanoparticles is quite complex. You have to control a lot of parameters. And of course, this leads to my last, one of my last slides referring to toxicity. Of course, there will be a lot of controversial about the toxicity of nanoparticles. As always, we can try to go to more biocompatible nanoparticles from the point of view of the composition, but we also need to take into account different parameters, because the size will be critical. If one nanoparticle is easy to incorporate and to deliver and to expulse from the plant, of course, they will not be toxic. So we have to think in different things, not only composition, but size, the concentration, of course, of using, the method of application, and also the plant. There are plants more sensitive to one type of nanoparticles to other. In conclusion, it's quite complex field. So I was thinking, how can we try to put some order into all these references that give some controversials or some very different results to a non-expert like me. And I think that probably machine learning and artificial intelligence can put some light here. There are a lot of studies performed in the literature in all the parts of the world, and I think that by using this useful information that are reported, we can try to incorporate it into a big database and try to make more reliable conclusions, I would say. So as a conclusion, I would say that nanotechnology is a promising area for exploitation in agriculture. The use of nanomaterials for seed treatment provides protection against biotic and abiotic stresses, resulting in improvements of productivity and food quality. The adoption of nanoparticle systems can alter crop management with reduction of the applied quantities of pesticides and contamination risk, resulting in more sustainable agriculture. This is safer for farmers, consumers, and the environment, but still remain some issues to be addressed, like the scale-up and low cost of the synthesis of nanoparticles as an industrial production, the seed priming conditions, to understand the toxic effects in plants and other organisms, but also it is necessary to improve the regulatory aspects. And I think this is all I wanted to talk to you, and I will be happy to answer some questions if.
Thank you very much.
Thank you.
Very insightful talk. I will, like I said, we have someone waiting. We had a problem that the people connected in Google Meet could not follow the presentation because we shared probably the wrong thing. But anyway, I think with the link of the YouTube channel, it was moving and they could more or less follow it. Unfortunately, we are very short in timing, but maybe we could have a question.
Thank you, Beatriz, for your very nice talk. And today I realized how complex and the possibilities of this field in agriculture, so it's very nice. But I'm very interested because I was in a meeting last week related to nanoparticles, and in medicine they are using a lot of lipid nanoparticles. And my question, I don't know if you are related to this field, but I have a question, maybe you can answer. Is that you think that this is a good opportunity or good method to use in agriculture, in seeds or in plants, lipid nanoparticles?
Okay, sorry. I say that I'm not a specialist, but I think that these systems are a little bit delicate because they are self-assembled structures that can be, I would not say destroyed, but altered because of many parameters like pH, presence of salts, and these kind of things. I think we have, under specific, we study very well the conditions in specific plants. I think there is a lot of room for that. Yeah, we use it in the creams every day. So I think it's possible. It's a nice way to a new address. Thank you.
Much. Thanks again, Dr. Loic Rajjou, for connecting, and thanks, of course, to Beatriz Julián for her outstanding presentation. Really insightful and really inspiring and encouraging to try to blend the new material science and the biological part.
Thank you.
Thank you very much. So next talk by Cynthia Bertea that Alma will introduce. We can decide what we want to do. I mean, we have questions in the YouTube channel. We can, for Adriano, Griffo, and also for Loic Rajjou, we can just ask them now, or we can just wait for after your talk. Whatever you decide. If Dr. Rajjou is in a hurry, we can just go on with the questions now. And
I can answer on the chat on the YouTube, or you prefer answer here?
I think it's better here. So, but you have time. Can you wait after the next talk?
I can wait. I can wait.
Okay, so please, Alma.
Go ahead and don't take.
Thank you very much.
Don't be later.
Alma, could you please introduce Cynthia, please?
Yes, Cynthia Bertea from University of Turin. She has a long-term expertise in the use of biostimulants for improving seed quality. So please, Cynthia, the floor is yours. Thank you for being here.
Thank you very much, Alma. So can you see my presentation? I share it. Alma, can you see?
Yes, it runs. Okay.
I told yes, please take the screen.
Yeah.
Okay.
It's okay, thank you.
Okay, perfect. So, since we have some technical problem. So, first of all, I want to thank all the organizer and Alma in particular for inviting me to this very nice workshop. I'm working on plant biostimulants, and I have done in my career some experiment also with the biostimulant used for seed priming. Today I'm going to talk a little bit to introduce what are plant biostimulants, maybe for the people they are not used to this kind of product. And then I will show you a couple of experiments and a couple of papers that we published a few years ago. But now we are going ahead also with some more research on seed priming. So very briefly, first of all, biostimulants are not fertilizer, of course, because the fertilizer as you know, are substances, mixture, or even microorganisms that can provide the plants with the nutrients, and also so it's not something that is just stimulating the physiological aspect of the plants like is doing the biostimulants. So the biostimulants usually are also natural product. They can be, as I will show you later, made by different matrices, but they have the particularity to stimulate the plants, so the plants does not receive nutrients for biostimulant, but all the physiological aspect, all the physiological processes can be actually stimulated by this product. So we talk about plant biostimulant when it's also stimulating the nutrient use efficiency, the tolerance, increase the tolerance to abiotic stress, also increase the crop quality traits, and also able to go to to give the availability of confined nutrients in the soil or rhizosphere. Plant biostimulants were quite a controversial topic in EU, and from the 2019, finally there is a regulation. So they enter actually into the fertilizing law, but of course with these very clear differences. So they don't provide nutrient to the plants. Very important into the biostimulants world is the EBIC, that is EBIC, that is actually a consortium that was founded in 2011, also by different companies, to foster the role of plant biostimulant in the sector of sustainable agriculture, with the motto more with less. So just by using a little amount of these biostimulants, the plant can have the capability to grow very well and to counteract also biotic stresses. As most part of you know for sure, so the biostimulants comes from very often from a circular economy, since we can use some residual wastes to prepare this kind of product. So the biostimulants are not anymore into the linear economy point of view, in which we have a production that is going to the end and we just have a wastes, but these wastes can become a new source for the production of interesting molecules. So the biostimulants are actually inside of the circular economy. So the circular economy will lead to the production of this kind of product that can be very important for the agriculture. When the companies are developing biostimulants, usually they think about the specific agricultural needs. So they make this kind of natural extract, but of course it is necessary always to have a clear idea of what is inside the extract, because since few years ago, this was not very clear. So they were just putting into the soil, into the plants, this kind of product without having an idea, clear idea about the molecules present inside of these mixtures. So, and once there is the desired combination of active ingredient, there will be the realization of a prototype, and then afterwards, also the measurement of the shelf life, the stability, and of course the safety into the field in order to avoid problems or environmental problems. Just very quickly, I will just give you an overview of what of a few different categories of plant biostimulants. Very often you can find inside of the biostimulants some algal products, especially microalgae, like for example Ascophyllum nodosum or Laminaria digitata. Then there are biostimulants also made by different kind of molecules, like a mixture of carbohydrate, proteins, amino acids, lipids, and so on. Some biostimulants were also coming from humic and fulvic acid that are present inside of the soil already, and other biostimulants can be made by the presence of microorganisms. So the presence, for example, of plant-growing PGPR, so the bacteria that are able to improve the plant growth. As you can see from this slide, the biostimulants have a different effect on the plants. They can be tailor-made for some organs. In other cases, that can give some activity on the whole plant. But as you can see, there is a marker that is quite common. So they also very important for the antioxidant activity. So they are able to counteract the ROS species in order to remove a bit, to reduce a bit the oxidative stress in the plants. So this is very important because, as you know, we have a very big problem with the climate change right now, and also this year we could see actually the effect of the climate changes, that by the year 2050, so we will have a big problem with the crop production. And of course, having big problem with crop production, this means also having problem in feeding the world population that is in an increasing way. So, as you know, the impact of the climate change are both biotic and abiotic, but in particular, for the abiotic stress, we have on the plants these different, let's say, these different effects. The stress responses, of course, leads to the production of reactive oxygen species in higher amount, reduce the photosynthetic activity, and reduce also the plant growth and the yield. Here I've just put few of the abiotic stress because actually when you also claim a biostimulant, is, for example, for the company, they talk more about abiotic stress than biotic. But of course, the effect of this kind of product is on both kind of stresses because also the stresses have a common pathway. So the biostimulants, they can be as I told you before, made by microbes and also by non-microbial matrices, or both of them. So the most famous are the application, the foliar application and the soil drench. And as I told you also before, so they have the capacity to improve, for example, the yield and the quality, stimulate plant metabolism, optimize nutrient use efficiency, and also have a good activity on the soil, especially for the microbial activity, and general protection for the plants. But what about at seed to seed level? So this figure, I think Alma and the group of Alma recognize because it's from one of their nice work on biostimulant. So we can use also the biostimulant at the very beginning of the life of the plants at the seed level. So in this way, we can prime the seeds with the plant biostimulants, and usually we get like an improved germination, also uniformity in germination most part of the time. And this everything goes to the final part of the plant growth, in which we have maybe an enhanced crop productivity, a much stronger stress tolerance, so a good stress resilience, and also an improved quality of the food. So the work I'm going to show you today is based on a biostimulant that is called KM, that I developed together with a company here located in Piedmont region, close to Torino. So I have a strict collaboration with them, and for the, let's say, the world of biostimulant, is very important to have collaboration with companies. And together we developed this kind of biostimulants, especially devoted to be active against heat stress. So the name of this biostimulant is KM, and it was developed with my PhD student, the company, and we have a kind of called apprenticeship PhD, in which the PhD student was working into the company and was working also at the university. So both of us have the possibility to work on this kind of product. So as I told you before, it's very important also to know what is inside of the mixture of a biostimulant. And first of all, what we have done was also the analysis of this plant biostimulant that actually is claimed to be based on lignin derivatives like lignosulfonates and also on plant-derived amino acid. So what you can see in this table is the composition of these biostimulants, especially from the amino acid part. As you can see, there are a lot of these of amino acid, also other, let's say, organic compounds that were just present in traces. But beside the lignin derivatives, that it is not very easy to analyze, we had also this kind of let's say, composition, amino acid. The first paper I'm going to show you is this one that was published in 2020 on soybean. Actually, this biostimulant was made for the Brazilian market, in which there is a lot of soybean cultivation going on. And as you can maybe know, for sure you know, that is okay, soybean is an important worldwide legume, but also the yield of the soybean is really affected by the heat stress at germination time. And due to the climate change, sometimes the germination, the germination is done when the temperature outside is about 35, 37 degrees. So this is important to try to help the seeds to survive to this strong stress. So what we have done in this work, it was a double way, a laboratory part and a field part. The laboratory part was done, of course, in controlled condition. And what we have done it was mainly focused at 35 degrees, because 35 degrees was actually the temperature that it was also recorded in Brazil, the year in which we start also our experimentation. As you can see, we just put the seeds into an incubator at 37 degrees in the dark. Some seeds were treated, others not treated. And after 48 hours, and especially at the 72 hours, we could see a good, let's say, percentage increase in seed germination due to the treatment with the biostimulants. With regard to the biometric parameters, we could also see some differences, because we use this smart grain software, and we could see that the area, the perimeter, the length, and the width of the seed were changed. They were increased after the biostimulant treatment. And what it didn't change too much, it was the weight of the seed. So after, let's say, 24 hours of incubation, we could see actually changes in the let's say, in the positive way, few of the parameters, but the weight was not really affected. So this was something that was quite a surprise, because we were thinking maybe if the seeds is also swallowy, maybe there will be also more, the weight will be higher, but it was not like this. So we hypothesized that maybe the biostimulants is doing a kind of mucilage around the seeds. That is, actually, the seeds will get less water than expected. So, and also the germination process will be a little bit changed, but not because of the high amount of water entering into the seed, but maybe more for gas exchanges. Okay. What we have done also, always by using this controlled condition, it was the biostimulant application that was also checked by RNA-seq analysis. As you could see, we also get some surprising results. These were done at 24 hours after the incubation. So we got really a lot of genes downregulated and few genes upregulated. But what it was very interesting that among these 52 genes upregulated, we could find a lot of methyltransferases and other kinds of genes encoding for proteins devoted to DNA repair, to RNA biogenesis, ribosome biogenesis, and so on. So some genes able to protect, let's say, as you can see from this table, able to protect all the DNA repair machine, and also to avoid damages related to the, let's say, to the possible action of ROS on this kind of important molecules such as DNA or RNA. So among the downregulated genes, it was also surprising because we got some responses that we were expecting maybe to be higher, but probably the way in which the seed was able to react to the coating, it was just trying to keep the damages of the nucleic acid lower, and so to speed up the to avoid the damages done by the ROS on this kind of molecules. We also check, of course, to the ROS, to the oxidative part. So we check the hydrogen peroxide along with some antioxidant enzymes. And also some other important molecules like non-protein thiols that can be able to reduce the amount of ROS. As you can see from this experiment, so the line, the dotted line is made by the control, that it was a stressed control. As you can see, we could see actually a lower amount of H2A2 in treated seed, and also the let's say the scavenger enzymes were also quite low, while we got a higher amount of protein thiols. This was what we got into the, let's say, laboratory work. Then we went ahead with the long-term effect on the seed treatment. So these experiments were done actually in Brazil, as I told you before. So the seeds were, of course, treated in the same way as we treated for the laboratory work. And then they were planted and they were sown, and then the plants came out. And what we had, we measured after 7 days, after sowing, we could see that there were some interesting parameters because, for example, the plant age was actually increased of the biostimulant treated seeds, also the root length, also the after 60 days, then the pod number. And the pod number is also very important, and also the number of seeds present inside of the pods was also increased quite a lot, and in general the yield. So this means that actually the treatment with this biostimulant can be really important for the final yield and also the health of the plants. These field experiments were done, as I told you, in the state of São Paulo, in Tacarituba, in which there is an experimental station. In conclusion, for the first work, we could just see that we had some influenced seed biometric parameters, also an increasing of germination. In this case, the germination was not faster, but it was evident the effect after 72 hours. And also we could see differences quite important at the level of the field, in which we could get actually good amount of seeds also, and good yield in general. And we had, of course, the reduced oxidative stress. The second paper is related to always to use this kind of biostimulant, but we use a cucumber that is a very easy model to work on the lab. We work mainly into the lab. What we have done, it was more or less the same protocol that we see for the soybean. We use in this case 28 degrees just for the normal temperature, standard temperature of germination, and 35 for the let's say heat stress temperature. As you can see, there is some differences, and the application of the biostimulants increased the germination of about almost 7%, and also the fresh biomass of the seedlings. So this was especially at 48 hours, in which we could measure actually all the parameters better than at 24. Afterwards, we also made the same experiment as I told you before. We could see also the effect of these biostimulants on non-protein thiol content and H2O2. And as we already observed for the soybean, the application of the biostimulants as a seed treatment decreased the content of this hydrogen peroxide. Much higher, the decrease was let's say, recorded at 35 degrees, so in the heat stress condition. This is quite typical for biostimulants because they are more effective when there is a stress going on. When you apply the biostimulant when there is no stress, most part of the time you see very little differences or even no differences. So the amount of non-protein thiol, also in this case, thiols, also in this case, it was really increased. So we confirm actually what we have seen into the soybean. We did more or less the same experiment. We didn't run RNA-seq analysis, but just a real-time PCR, so qPCR, mainly into the antioxidant enzymes. And also in parallel, we ran some biochemical experiments, so by looking at also at the activity of this enzyme. As you can see, at 24 hours, we could not really have such big differences, but at 48 hours, most part of the transcript were higher, so the transcript related to the scavenging system of the plants. And also, when we check at the enzymatic activity, more or less, we could see the same trend. So the trend was of course, was increased the activity of the enzymes when the seeds were treated and were incubated for the germination and the development of the seedling at 48 hours. We also checked an important enzymes of the gluconeogenesis, so the enzymes that is leading also to the production of sugars from lipids is inside of the glyoxylase cycle, is isocitrate lyase. We also checked the gene expression and the enzymatic activity. Also, the most interesting results were got at 48 hours, in which we could see from, let's say, at 37 degrees especially, we could see an increase of 1.5 times the transcription level, and at 30 plus 37 more increase in isocitrate lyase activity. Also here, starting especially from 35 degrees. Also, in this case, the conclusion were that actually the treatment also on cucumber seeds was quite good for increasing the percentage and the fresh biomass. We were just on seedling. It will be very interesting to see also at the end about the amount of fruits and so on, I mean, for the plants, but we just stop at the seedling level. Also, in this case, we had reduced oxidative stress in treated seed and an increased activity of isocitrate lyase gene and also enzyme and gene expression. So this means that the germination process was not touched by toxic effect or whatever, but it was improved by the treatment of the biostimulant. So what we can conclude by using the biostimulant, in this case we use just one biostimulant, but now we are also working on other kind of biostimulants derived from agri-food waste. So some biostimulant, one biostimulant that is derived from the beta-lyins degradation. So we would like also to use it as a possible coating or let's say priming agent. So what we could get it is in general that we can see the improved germination. Most part of the time the plants is also able to face better the environmental stresses and increase also the crop final yield. So I want just to close my presentation by some acknowledgements. So the greenhouse company R&D department, so Giuseppe Mannino, one of our researchers, and also Rumiana Karlova from Wageningen University, they collaborating to these two papers. And I want to thank you very much for your attention, and just if you have some question, I will be happy to answer.
Thank you very much, Cynthia, for your nice talk, and also thank you very much for your perfect timing adjustment.
Okay.
Very good, very good.
Yes, I try to run a little bit more because I know that we are a little bit maybe on delay, so.
It's perfect. So first of all, I would like to apologize to people watching this through the broadcasting on YouTube, because there's certain delay between when they post the questions and we actually see them and can ask the speaker to answer those questions. So that's first thing. So then, I don't know, here in the audience, if there's any question present, I will check then if we have some written question. I have a question. Maybe it's more like some food for thought. The improvement in germination, it doesn't seem to be very specific. I don't know, maybe other people in the audience present here or online maybe can also provide some ideas on this. That the improvement in germination does not seem to be specific of different priming treatments, but like a general trend. When you treat the seeds, they tend to improve germination. No, I don't know. What do you think about this? That would be my first thought about it.
I don't know if I get it, but in the case of plant biostimulants, we have a mixture of components, and we also speculate that maybe some of them can be actually more active than others into trying to increase the germination and so on. But okay, we are still working on that, because the best way will be also to have separate components to check and to see what is going on. In general, the biostimulants that you use for treating the seeds, actually they do this, they have this action of improving. But as I could see also before from other presentations, instead then speeding up the germination process, they really made, let's say, in the case of KM, it was at the later stages of the germination. So it was something a little bit different that is necessary to investigate. So I don't know if I got your question or if it's not like this. So
more like a,
yeah.
a comment rather than a specific question. Maybe the answer is that nobody wants to see a negative result, like
Yeah, sure.
a treatment that hinders germination or renders less productive seedlings or whatever. But still, I mean, it's puzzling. Well, I wouldn't say puzzling, but it's interesting to what you all have been highlighting for each from their own perspective, that we need to dig deeper in the mechanisms that deliver that improvement in plant height, plant productivity, stress tolerance, etc.
Yeah.
And I think that those contributions are really important and really decisive, just to remove this black box that Dr. Rajjou mentioned before regarding the priming effect, and shed some light on this whole world or field. There is any other question here? Yes, Dr. Rajjou has a question.
Yes. Okay, thank you. Very nice talk and very interesting room. I was wondering if you check the germination potential of the progeny of the next generation. No, you don't know if you have heritability of some.
No, but we are thinking to do that also because, as you could see, you also talked before about epigenetic thing. So this is something that we have in mind. Also with Alma, we talk every year when we meet at our congresses, and I think this will be actually something that we would like to do. There is a collaboration. There is a collaboration with one person from the agricultural department here in Torino as a bioinformatic guy, and also is very interested in this epigenetic effect. Will be for sure something very interesting. As I told you, for example, for cucumber, we just stop at the seedling level. For the soybean, we should collect a few seeds in Brazil and try to go ahead with these seeds. Yeah, to see if there is some inheritance.
Okay, I think it's a very good way. I think the transgenerational effect is a very open question. We don't know nothing about that and about biostimulants. I think it makes sense.
Yeah, absolutely. Thank you very much. Yeah.
Could you please? You have a question?
Just to comment that it's true what Cinzia says, that indeed when you go more in depth on the molecular mechanisms that rule the seed response to priming, to treatments, then you will find common players and also divergent players, and we need absolutely to fix this complex picture, because it is from this basic knowledge that we can get the right hints to establish, to better improve the treatments, the protocols of the seed technology itself. So it was just a comment. Thank you.
Yeah, in some cases you get even just an effect of more mitigation, for example, of the stress, than actually a real, let's say, reduction or a real action of as you could see, instead that maybe you expect that the ROS scavenging system will be actually upregulated, but it's not always like this. So sometimes the biostimulant is doing the other way, but is reducing for example, the H2O2. And what is also important is those biostimulants that I'm testing very often have phenolic compounds. They are antioxidants, so themselves maybe they can do already something in reducing the oxidative stress. Okay, sorry.
Much. I don't know if there's any other question. If not, we could just ask the questions that were actually waiting in the comments box. I've seen that Dr. Rajjou has already replied to the question. I don't know if you want to make some comments on essentially the questions were out of curiosity. The commenter says, thank you very much for the wonderful talk. And he says he's curious if there's an interest in checking the effect of bacteria-based priming methods on the cell wall of roots in seedlings or treated seeds. For example, if carbohydrates such as xyloglucans are secreted more in roots, contributing more to the positive charge in the spermatosphere. And you already replied, but I don't know if you want to make some comment. I think Loic Rajjou has already left, so he won't be able to reply. Hello.
Okay.
I was just wondering whether you already replied
Yes.
on the comment box. I don't know if you want to make some comments.
Yes. We didn't check the effect of microorganism on the seedlings and on the cell wall roots, but in the exudate we observe some enzymes from the plants, from the seeds, but also from the microorganisms, and many hydrolase and particularly protease, and some enzymes which are able to use carbohydrates like cellulase, like xyloglucan, like rhabdogalacturonane, sugar, etc., etc. So I think it's a very good question. So these enzymes are secreted by microorganisms, are able to hydrolyze protein and carbohydrate from the plant cell wall, and probably in our case, protein and carbohydrate in the cell wall of the seed tegument. And regarding xyloglucan, we found some xyloglucan in exudate in the spermosphere of germinating beans and cellulose. So I think that it's a good source of carbon for microorganisms. And probably it's selective. When I talked about modeling, there is many people that working on the modeling aspect of the dynamic of microbial communities, and they observe that there is some specific microorganisms which are able to produce a more efficient enzyme than other, which is a competitive advantage. And my idea it's we need to do more research on that in order to understand how we can select a very competitive strain of microorganism in seed priming or after during seedling establishment to protect the seeds and to be more competitive against the competition with pathogen. But it's a very interesting question by Conrado.
Very much. There's another question from Conrado Dueñas for Adriano this time. Sorry, we saw the question much later. So he's also praising your work. Could you please silence?
Yeah, sure.
The mic. Could you please silence the microphone? Thank you very much. He's also praising your work, and his question, let me see, is during your work with FOX1 assay, were there a bottleneck that you think will translate in the large-scale use of this method in the seed industry, and how do you think can be addressed?
Okay, so for now, our research activity just focuses on the laboratory scale, but for considering the economics behind or the price of the reagents and the working solution in general, we think it will be used for the large-scale utilization since the reagent and working solution are not too much costly. But for sure, this kind of analysis requires more experiments and evaluations. And but our evidence suggests that yes, it could be possible. And regarding how who will use this kind of technologies, I think both from industries or companies that needs for the utilization of a large scale or multiple times, and also for gene banks. So I think it can be adapted for so in the context of company, we can try to change working solutions. So working with less concentrated reagents to make the working solution, the test more economic friendly, as well as in the context of the gene bank. In the context of gene bank, we already assessed that it's non-invasive, but for sure we can evaluate the long-term effect of the solution, which was not evaluated. But for now, our suggestion is that, yeah, it could be addressed for both the gene bank companies, and it requires just probably some adaptation of the reagent, the working solution, but yeah, it may be used also for large-scale utilization in multiple contexts.
Thank you very much. Thank you for sizing. So just I will make a last question because I think it's very interesting and also contributes along with all that has been said, Cynthia as well as Eduardo, and just to clarify the aspects of priming, also making it a different world from what we call biostimulants, which are two different responses and are two different effects. And yes, I'm interested of that part of clarifying what the effects of each of the treatments have on plants or directly on seeds, and also how can we have a control, a direct quality control on the seeds treated that may also translate into a benefit when these seeds germinate and the plants are grown. So I don't know, you want to elaborate something on what you explained on how this can contribute to have a better treated or primed seed quality control? Thank you. My question was if what you explained with these different methods could be used to have a better treated seed quality control.
Sorry, you talk to me? Sorry, you talk to me?
No, my question was for Adriano.
So if I got your question, so you were wondering if these methods can help distinguish treated seeds, I don't know, can support the to the separation of distinguished treatment. Is it correct, or it was? So can you repeat your question just to be sure that I can answer?
Yes. So my question was if the methods you mentioned to do the screening of seeds could be used as a primed seed quality control, just to know exactly in which batches of seeds the priming had an effect, and whether these seeds will be working after the priming treatment or not.
Yeah, I think yes, especially the recent project or methods that we were going to develop with the volatile organic compounds detection. From our results, we observed that primed seeds are able to release specific volatile organic compounds which do not find in the other groups. So for sure, based on our preliminary results, we may distinguish the type of treatment, and I think it can be used for distinguish the qualities of priming or the type of treatment. This is based on our preliminary results, but of course we need further analysis and evaluation to be sure of that. But from a starting point, we think yes, because we observed, especially with the volatile organic compounds detection, different volatiles emitted in primed seeds compared to control seeds, which are also different to artificially aged seeds, which reflect that probably different processes are involved in priming or artificial aging or simply dry seeds. So yes, from our preliminary result, yes, it may be useful for this kind of distinction.
Thank you very much.
No problem.
So we are finishing the first part of our workshop. We will now have a break for lunch, and we will resume the workshop at 2:30. Thank you very much. See you later.
Thank you.
Thank you. Have a good lunch.